In this video, we're going to talk about the electrophysiology of the heart muscle. Now, the heart does not require conscious intervention to get it to contract. We don't sit there and think, now, heart, please contract. And we can't really do that. We can't control it directly. Now, we often talk about the heart having this autorhythmic behavior, that the pacemaker cells will cause a discharge, which spreads across the heart, and then we have the atria, then followed by the ventricles contracting. And that is true to a large degree. It really is rooted there. But what we'll see later on in this chapter is that there's a lot of external input, because our heart, of course, our heart rate can change depending on needs, and the force of contraction can also change. So there is outside input that is very important for controlling the functioning of the heart. Now, very much like skeletal muscle, we're looking at muscle cells that undergo polarization, depolarization, and also have action potentials. Now, let's have a look. When we're talking about the pacemaker cells, we're looking at specialized groups of cells that are located throughout the heart. And these cells set up arrhythmic, spontaneous generated action potentials that will spread to other types of cardiac cells, which are the ones that are responsible for the contraction. So we have these two different populations that are highly related: the pacemaker cells that are in certain locations and groupings, and then the rest of the myocardium, which are the contractile cells. Now, let's remind ourselves about the histology of cardiac muscle tissue, that we are dealing with striations. They do have striations, right? And that's because there's an arrangement of contractile proteins within them. Now, we also do have that similarity, physiological similarities with the skeletal muscle. That skeletal muscle and cardiac muscle have the same general function, and that is to generate tension through a sliding filament type of mechanism. Now, let's think back as to what these cells look like. They're branched, right? They're branched. They have a single nucleus. They're shorter and wider than your typical skeletal muscle cell. They have myoglobin. And of course, myoglobin has the ability to carry oxygen. And they're really dense, full of mitochondria. So mitochondria are very important because the heart really relies upon aerobic types of respiration. You don't want the heart to go anaerobic for too long. And so the mitochondria really reflect the high energy demands that we ask of this tissue. Now, if you think back as to the structure of the cardiac cells and how they're put together, we have these very special features, the intercalated discs that join the cells together. And of course, within the intercalated discs, we have the desmosomes, and they help to sort of staple the cells together. And very important, the gap junctions, and they allow ions to pass from one cell to the next and allow the intercellular communication. And in this case, in cardiac muscle cells, the communication is, hey, go ahead and depolarize. And just a quick sort of snapshot view is we can certainly see here are the intercalated discs. They're all over the place, and they mark the beginning and end of a cardiac cell. And we can see that, yes, we do have actin and myosin lined up there. We do have T-tubules. We have sarcoplasmic reticulum. Now, the role of calcium is a bit different here. We have a sarcolemma. Now, let's focus in down here at the close-up of the intercalated discs. And we can see that we have numerous gap junctions which allow the passage of ions between them. And we can see the desmosomes here that are really doing a good job to hold the cardiac cells together. If not, that would be a horrible situation if your cardiac cells detach from each other during contraction. So thumbs up for desmosomes. Now, like skeletal muscle and other excitable cells, the cardiac muscle cells have selective gated ion channels. And of course, it's the timing of the opening and closing of these channels that allow for things like polarization, depolarization, hyperpolarization. And we see that in both the pacemaker and the contractile cardiac cells, these channels are very important. So let's introduce these channels. Now, many of them are familiar to you from past chapters, is that we have the voltage-gated sodium ion channels. And of course, being voltage-gated, they open when there is a voltage change across the membrane. Now, where do we find these in the heart? We find them in all cardiac muscle cells except, and let me say that again, except certain pacemaker cells. They don't have this type of gate. We're going to see there's another type of gate coming up that we haven't seen before. We also have calcium ion channels. And they act like a voltage-gated opening. But it's a timed gate closing. So they close after certain periods of time, regardless of the voltage. So they open, and it's almost like there's a stopwatch going, and when time's up, regardless of what's happening in their local environment, they will close. Now, all types of cardiac muscle cells have one or more types of potassium ion channels. Some are ligand and other voltage, depends where you are. So there's some of the things so far. So we've seen that we have voltage sodium, we have calcium ion, we have potassium ion. Now let's introduce another one. And these are what we'll call the non-selective cation channels. And you'll see in your book it talks about the HCN. And a little bit about these. These are non-selective, found in pacemaker cells. And it's interesting, they're activated by hyperpolarization. So I'm going to use a marker on that, hyperpolarization. So that's sort of a unique feature that we haven't seen before, that hyperpolarization actually triggers something. But we're going to see this is really important because pacemaker cells, what happens is they set up their action potential, they fire, they repolarize, they actually hyperpolarize. And the fact that they hyperpolarize then opens these channels, and that sets the whole thing going again. That's why your pacemaker cells will fire on a regular time table, because they will never just sit there in that state where they have set up the basically the voltage difference like neurons do, or other muscle cells, where they just sit there waiting for an outside influence to tell them to go ahead and depolarize. And I've included this as a reminder. You know, voltage is the difference between electrical potential between 2 points. Membrane potential is the voltage charge that exists across the membranes of all cells, including these heart cells. And resting membrane potential is that membrane potential where we have excitable cells at rest. They're not being stimulated right now. And depending where we look, it's, minus 60 to about minus 90. And this is a result of difference in concentration of ions across a plasma membrane. Now, I'm not going to spend much time here. Current is the flow of ions. Depolarization is basically a change in the resting membrane potential to a less negative, to a less negative value than when at rest. And repolarization, of course, is the return to where it was. And we've seen those terms before, so we're not going to spend much time there. Now, of course, this is based upon concentration gradients of ions. So we have a concentration of sodium and calcium in the extracellular fluid, and it's higher than in the cytosol, right? Whereas if I look, the concentration of potassium ions in the extracellular fluid is lower than the cytosol. So that's sort of the situation that we have set up here. And sodium calcium ions will, of course, like anything, follow its concentration gradient if allowed to, and will enter the cell when we have channels, the appropriate channels opening. Potassium ions follow their concentration gradient and leave the cell when their channels open. And of course, not surprisingly, we have in the background things like the sodium-potassium pump that helps to maintain the gradients. We should also point out that calcium, because remember we did say calcium takes on a role here that we didn't see in terms of the skeletal muscle with it being outside coming in. It was stored inside. Really, that's where it was, in skeletal muscles. But here in cardiac muscles, we're really looking at movement of calcium from outside to in and inside to out. And the calcium ion gradient is separated by its own active transport pump. Now, pacemaker cells, they basically undergo a sequence where they set up an action potential, they hit hyperpolarization, they depolarize, and then they repolarize, and they keep doing that cycle over and over and over. Action potentials are transmitted from the pacemaker cells to the contractile cells through, yes, not shockingly, the intercalated discs. So those gap junctions really allow, once we get the ball rolling, for the ions to move from one cell to the next through those gaps at the intercalated discs. And of course, this allows the heart to beat as a unit, that the atria will contract as a unit. Then we get that delay, which we talked about in class because of things like the AV node and the presence of the fibrous skeleton will slow down the transmission of the signal to the ventricles. That allows the atria to contract while the ventricles are still in diastole and they can fill. So let's come here and start talking about the populations of pacemaker cells. Now, pacemaker cells really only make up about 1% of all the cardiac muscle cells. So, 99% of the cells in the heart are not pacemaker cells, they're run-of-the-mill. And we see three sort of population areas where these exist. And of course, the hallmark is that they can spontaneously generate action potentials. And this sets the pace for the heart. And the three cell populations are collectively called the cardiac conduction system. They're part of that. So let's have a look at this. Is the pacemakers act in a different way than your typical cells that we've seen to date. And they're different from those contractile cells. That depolarization, and this is going to be an important part, in pacemaker cells occurs more slowly. So it's not that big kind of explosive depolarization we see in things like an axon hillock from the past. And this is partly due to the lack of the voltage regulated, I'm going to highlight this, lack of voltage gated sodium ion channels in the pacemaker sarcolemma. So they don't have that huge number of sodium channels that we saw when we looked at something like an axon hillock. And the pacemaker cell's action potential lacks a plateau phase because the membrane potential keeps going back and forth and it never remains at resting level. As soon as it's at resting level, it starts a new phase of polarization, depolarization, that whole. Now, what leads to this? And it's something that we introduced near the start of this. It occurs because of the non-specific cation channels. And you haven't heard about these before because we don't see them elsewhere that we've come across until now. And because these really exist in the pacemaker cells. When we're looking at the pacemaker potential, what we see is we have a slow initial depolarization phase. So the pacemaker potential, how does it start this? The membrane actually enters a hyperpolarization state. And this hyperpolarization state, then what it does, it opens those non-specific cation channels in the membrane. And this allows sodium ions to leak into the cell and potassium ions to leak out. And the result is, it's not that big instantaneously depolarization. It's a slower, more drawn out depolarization. Okay, so the first part of depolarization is, let me say it again. The pacemaker cells are hyperpolarized. As soon as they get hyperpolarized, they open the non-specific cation channels. This causes sodium to leak into the cell and potassium ions to leak out. And I end up with a slow depolarization until I get to that magic word, which is threshold. There's a threshold in these cells. And when I hit threshold, that is when voltage-gated channels will open. And what type of channels are these? Calcium ion channels open. And this allows calcium to enter the cell. And this is what leads to the full depolarization. Now, repolarization occurs because the calcium ion channels, remember I said earlier, they have a stopwatch running. And they're a time-gated channel. So they're only open for anywhere from about 100 to 150 milliseconds. They close at the same time. And the voltage-gated potassium channels begin to open, allow potassium ions to exit the cell, and then the cell begins repolarization. After that, we have something called the minimum potential phase. And this is where the potassium ion channels remain open until the membrane reaches its minimum potential. Now, what does this mean? This means at the end of this phase, I am back to hyperpolarization. And of course, consider what that means. When the cell becomes hyperpolarized, who opens up now? It's the non-specific cation channels, and I start the whole thing over and over and over again. So it's a continuous cycle where there's really no big rest in between. I get the hyperpolarization. The effects of that is a depolarization. I hit a slow depolarization. I hit threshold. I get the real polarization, depolarization happening. I get repolarization. I hit minimum potential, which is hyperpolarization, and it starts over and over again. Let's just take a few minutes and let's just have a look at this diagrammatically. This is a great set of diagrams in your book, is that I get slow initial depolarization. And you can see the non-specific channels. When I'm in a hyperpolarized state, they open and I start to get the exchange of ions. And you can see I'm starting, from the depolarized state, and I'm starting to do the slow depolarization right here. Okay, so that's a slow phase. Then when I hit threshold, I'm now going to open these. These are being closed. So at threshold, I get the calcium gates opening. And here I see a much sort of active movement of ions. Calcium rushes in. And of course, calcium being 2+, I really start to see an increase in the membrane potential within, right? So we get that influx of calcium. Now, remember, these calcium gates have a timer, and they do close. So the calcium channels close, and the voltage-gated potassium channels open. And now potassium rushes out. So you can see, this is the point where the calcium gates have closed because they're on a timer. And now potassium gates open. And remember, there's much more potassium inside versus out. And we get potassium rushing out at this point. And we can see, because they're positive, they're leaving the inside of the cell. I'm becoming more negative. And then I actually overshoot. So you can see down here, I overshoot and I hit a hyperpolarized state. And of course, as soon as I hit the hyperpolarized state, HCN channels will open and I begin the cycle again. All right, we're going to stop there. That was pretty long. Make sure you get into the textbook, look at it. There are some videos associated with the notes you can see also on YouTube and also on our website. There are things for you to watch.